Well, good afternoon, everyone. We're so delighted you're here with us today. Welcome to Probable Meets Possible. The Bell Museum and the College of Biological Sciences launched this online series of conversations to bring together University of Minnesota scientists in bio-based fields to talk about the probable challenges we face and the possible advances that could have a profound impact on our future. These conversations, which you can find in a playlist on the Bell Museum's YouTube channel, have engaged audiences in exciting conversations about life in extreme environments, including other planets, the consequences of climate change on water availability and ice cover, and the business outlook for sustainable products and socially conscious investing. My name is Holly Menninger, and I'm the Director of Public Engagement and Science Learning at the Bell Museum. We are proud to be Minnesota's State Natural History Museum and a public gateway to research at the University of Minnesota. Our mission at the Bell is to ignite curiosity and wonder, explore our connections between nature and the universe, and create a better future for our evolving world. Through our Probable Meets Possible series, we've engaged you and an excellent lineup of researchers in conversations that explore big ideas and new directions in science, that allow us to engage each other in some future thinking about what lies ahead for our health, for the environment, for the planet, and in today's case, with respect to microbes in the environment. We are grateful to our partners in this series, the College of Biological Sciences, led by Dean Valerie Forbes, and the CBS Communications team, including Stephanie Zenos and Claire Wilson, in addition to our own Amber Kastner and the rest of our own Bell team. Thank you. Now let me introduce our special guest today. Dr. Will Harcombe is an associate professor in the Department of Ecology, Evolution, and Behavior. Dr. Mikael Elias is an associate professor in the Department of Biochemistry, Molecular Biology, and Biophysics, as well as in the Biotechnology Institute at the University of Minnesota. Our topic today, learning to speak microbe. This afternoon, Will and Mikael will take us on a tour of the state of their science and share insights about the ability of microbes to communicate, the effects of this communication on the environment and human health, and recent and future advances in the understanding of the numerous microbial languages. We'll have brief presentations from each researcher and then open up the floor to conversation. A few technical notes. Please send in your questions or comments directly to the panelists through the chat box. Our team will be monitoring and curating those for conversation. You may also have noticed that we've turned closed captioning on. If you find that distracting or not useful, you can hide it by clicking on the live transcript button and hitting hide subtitles. We are also recording this webinar and we'll post it online to the Bell Museum's YouTube channel. Okay. So I think we're about to get started. We'll turn it over first to Dr. Will Harcombe from the Department of Ecology, Evolution, and Behavior. Will, all yours. Why don't you turn on your mic and share your screen?
Great. Well, yeah, I want to first thank the Bell Museum for organizing this. The Bell Museum is such an incredible resource that we're so lucky to have around. And so it's always great to engage with them whenever possible. Let me hide this. Okay, so I just wanted to start us off with this image to really highlight how beautiful and incredible microbial systems can be. So this is an image taken by Dr. Mark Welch showing a example of microbes actually from plaque associated with teeth. So you have streptococcus around here in the green and the outside, and corynebacterium here in pink, spandrels in the middle. And I just wanted to start here to highlight that this fundamental amazement of biology is really a lot of the reason that many of us get engaged in studying science. But in addition to wanting to understand the complexity and the beauty of the world, obviously microbial systems also have lots of other important applications. So for example, you all are really familiar with thinking about how microbes can influence your health. I think we're all acutely aware of that, especially at the moment. But in addition to influencing our health, microbes also have really important implications for global nutrient cycling. And so as we try to confront some of the challenges of a rapidly changing world, it's going to be critical to understand how microbial communities are contributing to that. And finally, microbes are these incredible chemists. And so there's a lot of excitement about trying to harness some of the power of microbial chemistry for a whole array of applied processes, from generating novel fuel sources to breaking down some of the pollutants that we're releasing into our environment. And so my lab is really excited about trying to understand the composition and function of microbial communities, to predict how the dynamics of these systems, so how the composition and function will change through time. And then ultimately, we'd like to develop tools that will help us to manage these properties, these emergent properties of microbial communities. And one of the things that we're discovering as we dig deeper into microbial systems is just how incredibly social microbes are. So microbes readily communicate with each other, and that's a lot of what we're going to be focusing on today. And Dr. Elias will sort of dig more directly into some of the incredible ways that they communicate specifically. But also microbes are incredible systems for studying cooperation. So they readily generate public goods, and they've actually helped us understand a great deal about sort of the origins and the stability of different types of cooperation. So game theory is really tightly integrated into microbial studies. Additionally, microbes engage in all sorts of conflict. So they engage in chemical warfare through releasing antibiotics that kill other strains. They even engage in biological warfare. So these cool cases where microbes, bacteria, can come into an environment and then, while harboring a virus in its genome, when it gets into that new environment, it can release the virus into the genome. That virus then sweeps through and wipes out resident competitors so that the invading bacteria can come in and take over an environment. And most recently, there have been these cool studies where they've also shown that bacteria engage in toxic knife fights. So they have this protein that they've co-opted, and they'll jab it out of their cell into any cell that comes up next to them, and can eliminate competitors that way as well. But one of the most fundamental ways that microbes interact is through modifying their chemical environment. So for example, if one microbe or bacteria consumes a metabolite out of the environment that another could use, that can lead to competition. Or if one secretes a product that another can use, that can lead to facilitation. Or as we just mentioned, if one releases a toxin, that can lead to inhibition of another species. And so in my lab, we're really excited about trying to take a bottom-up approach to understand the complex composition and function of microbial systems. And so ideally, what we'd like to be able to do is start with a genome sequence of a single species and use that genome sequence to understand the physiology of the cell. So what can it eat? What cool chemistry can it carry out? What is it likely to spit back out into its environment? We then want to understand how this intracellular physiology leads to interactions between species. And then ultimately how these species interactions drive community properties. Furthermore, because microbes can evolve so fast, we want to understand how selection at each of these levels of biological organization feeds back to change the genomes that encode life. So some of the approaches we take are starting with computational approaches, where we take genome sequence and use that sequence to create genome scale metabolic models of different bacteria. These models incorporate all known metabolic genes that are encoded in a cell and the chemical reactions that those genes carry out. Using these models, we've then developed a computational platform that we call COMETS, for computation of microbial ecosystems in time and space. So we can seed biomass into an environment, and then through simulating the activity of these metabolic networks, we can simulate the growth as well as the chemical gradients that arise on a surface. And in this way, we can quantitatively predict how both the composition of a microbial community as well as the function of that community will change through time. We then pair these computational simulations with experiments in the lab, where we take strains that interact through known chemical pathways, and we then engage in experimental evolution. So we can set up many replicates of these communities. We can track the dynamics of all the species through time, and not only track ecological dynamics, but also watch evolution happening right in front of our eyes. And so often what we do is simulate to come up with expectations, test that in the lab, find out just how wrong we were, and then use these lab findings to improve our ability to simulate and predict in the future. So I'm going to give you two just very brief vignettes of some of the projects we're working on. The first one has to do with thinking about applications for human health. So cross-feeding is one of the chemical interactions that we often see in the microbial world. And cross-feeding is when one microbe obtains metabolites from the excretion of another. And indeed, these are super common in the human microbiome. So for example, Ryan Hunter over at the medical school here has shown that Pseudomonas pathogens in the lungs of patients with cystic fibrosis cross-feed off of anaerobes that exist in the lungs. So the anaerobes break down the mucin, the mucus that's in the lungs, and these other pathogens rely on that mucus. Similarly, other researchers have shown that complex networks of cross-feeding interactions occur in the gut and are really critical for the degradation of metabolites in the gut. So we're interested in thinking about how these chemical interactions between cells, this metabolic communication, if you will, might influence the different types of treatment. So specifically, we were interested in thinking about, well, what would happen if we had any individual species of bacteria and we looked at its ability to respond to stress or survive a stress? And how might this stress change in a complex community where all three species were interacting? And could we predict what would happen in this three-species scenario from the single species dynamics? So we tried this with 3 strains of bacteria, E. coli, Salmonella, and Methylobacterium extorquens. What you're looking at here is the top-down surface of a plate. So we have an antibiotic disc right here, and then our bacteria are fluorescing. And depending on how resistant or how tolerant of that antibiotic they are, that determines how close to the disc they can grow. So you can see here that our Salmonella is super resistant and grows right up to our antibiotic disc. And in this case, we are applying tetracycline as the antibiotic. But our E. coli and our Methylobacterium are much more sensitive. So the question is, what then happens in the community scenario? And what we ended up observing is that when all 3 species engage in mutualism, so they cross-feed off of each other, that dramatically changes the tolerance to antibiotics. So even though Salmonella is super resistant on its own, in a 3-species mutualism, it becomes much more sensitive. So this was work carried out by Dr. Beth Adamovich when she was a grad student in my lab before she moved on to bigger and better things. And she came up with the term, the weakest link hypothesis, to describe this. So if you're in an obligate mutualism, your sensitivity is set by the most sensitive species for the weakest link within that community. We then went to interact with the Hunter lab over at the med school here at University of Minnesota. And as I just mentioned, they've shown that in the lungs of cystic fibrosis patients, there's a very similar cross-feeding interaction where the primary pathogen, the Pseudomonas, will cross-feed off of metabolites released by anaerobes. And so we did a similar test where we looked at the resistance of the pathogen, in this case, the Pseudomonas, to ampicillin. We found that at our highest concentration of antibiotic, there's no significant reduction in the growth of the pathogen. However, the anaerobes are highly sensitive. So even our lowest concentration of antibiotic led to a dramatic reduction in anaerobes. then consistent with what we observed in the lab, we found that in a cross-feeding scenario, we were able to inhibit this primary pathogen with dramatically lower concentrations of antibiotic. And Dr. Hunter is now going on to use this information to think about better ways to develop treatment strategies for patients with cystic fibrosis. Another vignette, which I'll just touch on very briefly. This is work that we're just starting to look at signals in the soils. So a great deal of work has demonstrated that bacteria in agricultural soils have a diverse array of impacts on crop production. So for example, they can produce antibiotics, which kill pathogens. And additionally, they can produce compounds which actually facilitate the growth of crops. And so we're really excited in a collaborative grant with colleagues both here and in the UK to think about how we can manage some of these signals in the soil. And this is work primarily driven by Dr. Linda Kinkel. And she's, for many years, worked on specific microbes from the soil called Streptomyces. And she's mapped out the diverse ways in which different strains of these Streptomyces signal to each other, and how these signals then influence the production of pathogen-suppressing antibiotics by different strains. And what she's been able to show is that by mixing the correct combination of Streptomyces into soils, you can dramatically increase the production out of agricultural soils relative to untreated controls. And so what we're doing now is combining some of our computational tools to make some quantitative predictions about how the chemical signatures of the soils will change through time. So with that, I hope that I've given you a flavor of some of the ways that we're trying to take a bottom-up approach to understanding microbiomes, linking genome sequences to microbial community dynamics, with metabolites being a key mechanism of communication within these systems. And I just want to thank some of the incredible folks on our team. And with that, I'll hand it over to Mikael.
Thanks, Will. Before we hand it over to Mikael, I might we'll do a little question in the transition.
Okay.
Had me at toxic knife fights among microbes. When one of your slides. Tell me a little more about that microbe that stabs the other. That was fascinating.
Yeah, so it's called a type 6 secretion system, and it's been documented in lots of different types of bacteria. But one of the classic examples is in Vibrio cholera, actually. And it's a way of sort of recognizing kin. So it's a protein that they co-opted from a virus that they stab out of the cell. And if there's another competitor that sort of grows up next to them on a surface, then it jabs into that cell and releases toxins which kill the other cell. Yeah, and it can create these really pretty patterns where you get sort of stripes of different types of bacteria. And yeah, I think it's a pretty exciting mechanism of interaction that we're just starting to understand.
Yeah, there is a whole hidden world of microbes, which hopefully we will kind of get into exploring more. And I'm going to encourage our audience that as you have questions, I know that a few have already entered the chat box for Will, so we'll get to those when we get into the larger Q&A. But I want to encourage people, as questions emerge throughout these talks, to please put those in the chat box, and that we will incorporate those into our conversation. So thank you, Will. We will be back with you shortly. Mikael, it's all you.
Right, thank you. Good afternoon, everyone. Thank you very much to the Bell Museum for organizing this. This is wonderful to share with you guys some of the things we know and a lot of the things we don't know about microbes. And I think that Will's talk was actually awesome for me. It's going to make my life easier because he talked about all of the types of interactions between bacteria, and there are so many between corporations and stabbing. And I'm going to focus more on certain exchanges, chemical exchanges between bacteria that we name communication and languages. And some of the work we're doing in my lab in particular, where we try to learn how to interfere with these communication systems and perhaps how to learn these languages. So I'm going to start to give you a very broad and wide view of the problem, and then I'm going to go into some of the fascinating prospects we have. So microbial signaling really is about bacteria producing chemicals in the environment, secreting them, and then being able to sense them. And one very classical mechanism for this is called quorum sensing, because the concentration of signaling molecule in the environment is actually a function of cell density. So it's a cell density dependent mechanism. The more bacteria, the more signaling molecules you have in the environment. This is actually a way for bacteria to count themselves and to coordinate actions that otherwise would not, like actions that taken individually would not be beneficial, but are very beneficial when taken collectively. And we're going to review some of these. So when you have a certain cell density, when you reach a certain concentration of these signaling molecules are capable of diffusing inside the cells. They are bound by cellular receptors, and they activate the expression of genes in bacteria. And this can account for 5 to 25% of the total genes in a bacteria. So it's a very, very significant mean of changing behavior. And in fact, this allows bacteria to adopt group behaviors. Group behaviors that include, for example, biofilm formation and virulence factor expression, which is of major interest in human health, obviously. Here is an example that is a lot more benign and more fun than biofilm or virulence. This is a case of a Vibrio strain, Vibrio fischeri. It produces bioluminescence in the lab. This is how a flask of it could look like. And this bacteria is performing a symbiotic relationship with an animal called bobtail squid. And this bobtail squid is hosting this bacteria on its bottom part here. As you can see, it's fluorescing at the bottom. And in exchange for nutrients that is provided by the squid, this bacteria produce light. Now, why is this light useful to the squid? It's useful to the squid because as the squid swims in the ocean, it's actually casting a shadow at the bottom of the ocean from the light produced by the moon. And this shadow can be used by predators to locate the squid and eat it. And so these bacteria on the belly of the squid are producing light that is actually compensating for his shadow and make it invisible to potential predators that are beneath it. So this is a pretty cool example of what cell communication can do. Now, what we do in the lab, and this is really microbial language 101, is that what we can do once we know that bacteria communicate using small signaling molecules, the first thing you can think of really is, how do I stop it from happening? How do I break this communication system? Because as you know, breaking is a lot easier than making. And so really, this is the simplest thing we know how to do. And you have these bacteria, they produce these signaling molecules. We know they use it to coordinate a variety of actions. This is the type of molecule they use here. The type of molecules they use, they're called, in this case, acetylhomoserine lactones. There is a variety of them. They vary by this R group. This R group can be very short, very long, can have various decorations. So they produce these molecules. What we can do is to intercept these messenger molecules, intercept them by many ways. The way we use in my lab is to use enzyme that degrades them. So we degrade these molecules. This is essentially a blurring device. So you could think of something that would blur the surface of your mobile phone. This is what we're doing here. Now, once you do this to bacteria, you in fact prevent or inhibit all of the bacterial behaviors that depends on this communication system. And I mentioned earlier that biofilm and virulence are mechanisms that depend on this communication system. So some of the work that my lab is doing is to study model systems in which we can verify that this is really happening. So we have some animal models here where we protect, here it's a worm called C. elegans. It's a millimeter-sized worm, very small worm that we can infect with various pathogens that can be resistant to antibiotics, for example, and we can verify that by disrupting microbial communication, we can protect these animals from death or here from being paralyzed by the infection. This is a similar experiment that was conducted with mice by our group and collaborators a few years ago. And then we have a whole sorts of other fields of applications where this time bacteria are involved in biofilm formation. And these are some of the more recent studies that we conducted in the state of Minnesota, and now we expanded in the Gulf area, where we can inhibit more complex behaviors where bacteria are key. So we saw earlier that bacteria were key in some behaviors in health, for example, but they're also key in more complex processes like biocorrosion or biofilm and biofouling formation. So for biofouling is everything you can see on the hull of a boat, for example, or everything that looks green on the glass of an aquarium. What's green are algae, obviously, but these algae often require a layer of bacterial biofilm before they can adhere to a surface. And so all these complex biological processes, very often they depend on bacteria. And so if you significantly alter the natural course of event and prevent a large group of bacteria to communicate, you will also affect these complex behaviors. And so we see that by quenching microbial communication, we can, for example, inhibit biocorrosion. This is the control, this is the treatment. And the same thing here with biofouling, where we can significantly reduce biofouling here, including the adhesion of zebras here that is present. We significantly reduce them here. So this is really 101 in terms of what can we do once we know microbes communicate. And from there, I'm going into more into perspectives and where we would like to go in the future. The importance of microbial languages is obvious. We have all heard about the importance of health-related bacteria, microbiome, gut microbiome, how to get a healthy microbiome, that having an unhealthy microbiome is linked to many diseases, etc. Now, the question really that matters is, once we know this, is how do we go from an unhealthy microbiome to a healthy microbiome? This is really the question now that is pressing. And as Will mentioned, there are so many things going on. There is the competition for food, there is the cooperation, there is the competition, and there is communication using signaling molecules. And so how do we go about this communication? And how do you go from an unhealthy state to a healthy state, or prevent a healthy state to go to an unhealthy state? So what we do know is that some bacteria produce and or respond to many different molecules simultaneously. And so this already increases the level of complexity very significantly. So it's not just one bacteria, one molecule. It's a lot more complicated than that. Some bacteria can produce a lot more than one molecule. Some bacteria can recognize a lot more than one molecule. Some bacteria can respond to a lot more than one molecule. And so essentially what you're dealing with is this kind of situation. So you have a whole lot of bacteria coexisting, and some of them are fighting, some of them are cooperating, but all of them are talking. And they're talking in very different languages, okay? And really our objective, ultimate objective, is to get an idea of how does that work. So this is a very similar picture. You have all these bacteria. Some of them are nice, some of them are less nice to humans. They're all talking. You have a variety of signaling molecules in the environment, very high number. And how do we go about that? So I'm going to here attempt an analogy with language, which I like to use in my thinking and in my experimental design. And so if we consider that different bacteria can produce different signaling molecule, which is actually the case. So here, if we take an example of this blue bacteria making this blue molecule and this brown bacteria making this brown molecules, and we assimilate these molecules as letters, we can think that there is actually a microbial alphabet, because the collection of these molecules is very, very large. Okay, and you can note that these molecules, they all look alike. They all have what we call here a lactone ring, and then they have an amide bond, and then they have a tail, and that tail vary as a function of the microbe, as a function of the bacterial species. So if you consider that this is an alphabet, which is the hypothesis that we're going with, you also can see that some of these bacteria respond to numerous of these letters at the same time, simultaneously. So it's, again, I'm repeating this, not all bacteria are recognizing only one molecule. Some bacteria can do many simultaneously. And so this means that if you have a complex community, you will have many of these letters mixed together. And you can imagine that these letters can form words, and that bacteria can recognize these words because they can recognize and respond multiple of these letters at the same time. Now, there is another level of complexity that we know of, which makes it even more challenging, but also even more interesting to us, is that not only you have many different letters, not only bacteria can recognize all of these letters or many of these letters, but also the relative concentrations of these letters in the mixture vary. And bacteria will respond differently whether the concentration of this molecule here is low or whether the concentration of this molecule is really high. And so this extra level of complexity, you could think that it's similar to making sentences with these words. And so here I give an example of this molecule is present, but its concentration is so low that this other one takes over and change completely the meaning of that word. Okay, so we're dealing really with a very complex situation where bacteria have elaborated very sophisticated ways of communicating. And this is not the only language that we know of. We know that there are other languages, and we know that there are other alphabets. Alphabets means that this is another family of communication molecules, and these also vary. So you have also a lot of variations of these type of molecules. So really we're super excited by these ideas of understanding the meaning, the biological meaning of each of these molecules, And in particular, what we're really looking at, hopefully soon, but it will take us some time to establish the first dictionary. So this is really one of the objectives. Learn one microbial language, and perhaps in the future, one day, we should be or will be able to speak this language. And if we can speak one of this language, that possibly means that we will be able to control microbes instead of fighting them, and perhaps even hacking them, because if you think of a language, you can think of creating a language virus, pretty much the same way informatic viruses work. So we're really hopeful that this idea can have success where the current paradigm is failing. The current paradigm being we treat microbes by systematically trying to wipe them out, either by using disinfectant or by using antibiotics. And this has been pretty successful, but it's currently failing due to overuse and adaptation of microbes. And so this is a possibility where instead of wiping them out, we might be able to control them through communication and solve some of the major problems where bacteria are involved. And that means numerous industrial issues and fields, food security, as well as human health. And, of course, I would like to thank all the people in my lab that inspired this by their work and my funders. Thank you.
Thank you, Mikael. Much appreciated. Before we transition to conversation, when we bring Will back, I'm going to ask a follow-up question. Is quorum sensing something that all microbes or all bacteria can do? Is that a universal property of those?
Yeah, this is a great question. It is unknown. We know that not all microbes are using the language that I presented. This is known for sure. We know that some microbes are using this language and also using another language simultaneously, so they are multilingual. We know that some microbes are known not to use any languages that we know of, but perhaps they have another language we have no idea of. So as of now, I personally see this as having all these microbes living in the same room. And I have a hard time thinking that some of them speak and some of them don't. Mm-hmm. And so I do personally believe that all of them do speak, and that we have a lot to discover from studying them.
Fantastic. All right, Will, come on back to us. We're gonna have a conversation. We've had a lot of great questions come in from the audience, and I encourage people to keep those coming. Maybe the first one that I will kick off for you is that how did we come to know or understand that microbes were speaking languages in the first place? Like, tell me a little bit about that. Like, how long have we known this? How did we find it out? How did we happen to overhear what was going on? I'm curious about sort of the history of and depth of our understanding.
Well, do you allow me to start?
Please.
Okay. So some of the first findings about this phenomenon really comes from the visualization of bioluminescence. And so people were wondering, how does it work that bacteria can make bioluminescence? When they actually cultured this bacteria, they realized that they don't form bioluminescence all the time, but they only produced bioluminescence when they were reaching high cell density. Really a high number, if you wish. And so this is where this idea of cell density dependent communication came from. And therefore, the discovery of the first molecules that are regulating the system of communication. And that was about 40 years ago. Yeah.
And did it inspire? I mean, so it happened. So with bioluminescence, and then did it just inspire people? Like, I'm going to look in soil. I'm going to look in the gut. I'm going to look like it just kind of grew from there.
Exactly, and then people started to identify the genes that made these compounds, and then there was the explosion of genome data, and people found these genes are actually widespread in a lot of bacteria. So a lot of bacteria make these molecules, and so people realize, well, my favorite bacteria also makes this. So what does it do with it? And it propagated like that. And I would say there is really a fine line and a difficult line to draw, and maybe Will can have an opinion on that. I have a personal opinion that it went even too far. Like, people were finding signaling molecules everywhere. And in essence, and the definition of a signaling molecule is very difficult to establish, because bacteria will virtually respond to almost anything you can do or add to their media. And so if you just define a signaling molecule by a molecule that will trigger a response by bacteria, literally everything is a signaling molecule. Glucose or any antibiotic would be the best signaling molecule, most likely. So really, those signaling molecules, they respond to criteria that, first of all, they are synthesized by bacteria. And second of all, they trigger the response of large portion of genomes to coordinate group behavior. So this is really something that is central here.
Yeah, I think that's a great point. And I sometimes like making the distinction between a signal and a cue, where a signal is something that has evolved to be released specifically because of the adaptive benefit of the response in a receiver. Whereas a cue might be something like, oh, the glucose in my environment is decreasing. I should start producing antibiotics to kill off a competitor or something like that. So you can have sort of this response to another player or another species that I agree with Mikael wouldn't fall under what I think of as a signal. But,
yeah. Related to that, what somebody in our audience wanted to know, and I think this is kind of related to what you're talking about, is it known whether these signals are only used for communication purposes? So the chemicals that they make and release, or are they also used in other pathways?
I can talk for the ones that I know best. This is a great question again, and one of the things that I can tell is that we don't know for sure, but we know that these molecules, some of them do show some cytotoxicity. So that means that some of them are toxic or killing some bacteria. So there is the question, there is also the question there of their origin. How did this communication, this very complicated communication system evolve, and from what? Because you think that you have to evolve synthetase to make these molecules, but you also have to evolve the response component. And one of the hypotheses is that these molecules were actually ancient antibiotics that were so widespread that bacteria adapted and evolved a systematic response to it. And they kind of lost their ability to be an efficient antibiotic, but the communication and response mechanism remained. This is just an hypothesis. There is not a lot of evidence for it. But another role that some people have found is that some variations of these molecules are involved in ion uptake. Some variations of these molecules in particular are iron siderophores. And iron is a very key nutrient for bacteria. So this may be still a very important role, or may have been a very important role in the evolution of these systems.
Yeah, and following up on that, we in some of the signal in the soil work, we have ongoing debates about what really is a signal, and there are people, like Mikael was saying, who argue that actually most antibiotics are just signals, that in the concentrations they're naturally produced, they're not killing other microbes most of the time. They're instead just transmitting information. I have a hard time with that, and I think most of the time they are weapons as opposed to signals. But there's definitely a gray area there where it's unclear. Yeah.
But what is clear though is that antibiotics is an extremely strong selection pressure to evolve a response mechanism. Yeah. Because there are only two options with antibiotics, is that either you evolve a response mechanism or you die. And so in that respect, there is the argument it's a signaling molecule, right?
Right. Yeah. I mean, depending on your definition, yeah, it's definitely true.
So one of the things I thought about during both of your talks, you were kind of talking about looking at like the genes that are turned on, and so I'm wondering, how have advances in genomic sequencing, I mean, I've seen it sort of in just over a very, very short period of time, how has that sort of revolutionized or impacted your ability to do your work? And I'm assuming led to kind of a whole new field of questions, things that you would not have been able to answer if we didn't have the sequencing technology that we have. Maybe you could talk a little bit about that.
Yeah, I can start. So first of all, like the availability of genomic data, like I said earlier, has allowed us to see what is the fraction of bacteria that, do use these systems. It has allowed us to see that some bacteria make these compounds, but don't have the receptor for it. So they make these compounds, but they do not respond to it. So it's like they're capable of speaking, but not hearing, for example. So you have very interesting variations there. Or some bacteria do not have these systems, but they have a brewing device for this system. But also, personally, in terms of research, the availability of deep sequencing methods have allowed my group to study the effect of signal disruption, the effect of interfering with microbial community on the microbial population itself. And so we have seen that when you actually suppress these communication systems, we significantly alter the microbial population in a given environment. And we're still studying this. We're still trying to understand how these changes occur. Our working hypothesis is that when we suppress communication, we alter the fitness of certain groups of bacteria. And this changes in fitness lead to change in population structure that we are observing.
Yeah, I mean, it's definitely sort of building on that. I think sequencing has given us this amazing insight into the parts, right? So we know the parts that make up a single cell, all of the genes that combine to encode a single cell. And we know now all the species that make up a community. And I think with the advent of these sequencing tools, actually, the sequences have outpaced our ability necessarily to make sense of them. And so that's what, our genome scale metabolic modeling, that was one of the real drivers, is we have these mountains of data on genome sequences, but how do we convert that sequence data into biology? And so we're starting with the metabolism and then hoping to get to the point where we can incorporate some of these signaling molecules that Mikael has been talking about. And then the next great revolution is going to be that we not only now have the ability to read genomes, but we also have the ability to write genomes. Mm-hmm. So with the explosion of CRISPR-Cas, can we get to the point where, sort of like Mikael was suggesting, we can program microbes to carry out specific functions, to communicate in desired ways. And Yeah, I mean, it's an incredible time to be a biologist with just the amount of data that we have at our fingertips, and the amount of tools that are coming online.
Speaking of writing genomes, somebody asked early on, somebody named CK for Will, and I think that's related. Would you describe some of what you explained as synthetic biology? We had the last time we had a probable possible, we were actually talking about synthetic biology.
Nice.
And so where's the line there? Do you consider, like, as you were saying, like writing genomes, do you consider any of the work you do in that space?
Yeah, definitely. I was actually hired on a synthetic biology job announcement. So yes, I definitely think I'm doing some of that. I guess the way I think about doing it the most is maybe synthetic ecology. So combining, identifying combinations of microbes that will be really effective at suppressing pathogens for farmers in Minnesota, or that we can sort of manage probiotic treatments for humans to modify the gut environment. But that's another real power of some of our modeling approaches is that they're just totally made for synthetic biology. So we can throw in novel gene cassettes and say, if we added these genes to this bacteria, how would that change not only this bacteria, but the entire community? So I think there's all sorts of really exciting potential there. And it's definitely a direction that we're headed. Can I jump in with one of the questions from here?
Yeah, you bet.
Someone was asking about viruses making signals. And Mikael, I know some of that from phage, but are any of the homoserine or the ACL classic quorum sensing molecules, have they been found in viruses?
Not to my knowledge, but okay. Now you have this finding with giant viruses that have really large genomes, and I wonder whether we will actually find this pretty soon. But I don't know of it.
Okay. So with some of the prophage that will integrate into bacterial genomes, there are some sort of pretty recent studies suggesting that those viruses can communicate through just a totally different language, and that can actually influence when they basically pop out of a cell, so when they excise themselves from the genome and burst out. It seems like it has some correlate or relationship to quorum sensing, but as a totally unique language, like you were saying.
Yeah.
It's surprising though that you wouldn't see the classic quorum sensing ever integrated into viruses. Because it's not that big, is it?
It's not that big, but there is a lot of pressure to keep most viral genomes really, really small.
Yeah. No, that's true.
But these giant viruses, they really have big genomes, so I would be surprised that we can't find something like that there.
Yeah. Huh.
I want to kind of go into the realm of potential therapeutics, and there was some interest from our audience there. So, for example, someone asked, does drinking something with probiotic properties like kombucha introduce microbes that speak a new language or maybe enhance the language that would improve gut health? Have thrown that one your way.
I have an opinion. No, I mean the efficacy of probiotics in humans is very much controversial in the literature. You have actually relatively few papers showing there is much effect. It is almost a philosophical debate, I think, in microbial ecologists. There is this debate whether what matters is the microbe or what matters is the milieu. And I personally believe that the milieu is most important. So you can add all the microbes, all the good microbes you want. If your milieu is not appropriate, it will not stick. However, there is one example, which is not exactly probiotic, and it's a very specific situation, But there is one example where clearly the exogenous addition of a microbe is very beneficial. It's the case of fecal transplant, and the work, for example, from Mike Sadowski and Alex Koretz at the UMN for healing of C. difficile infections. And so this is a case where C. difficile took over the gut microbiomes because of use of antibiotics. And then it's very, very difficult to remove. And the fecal transplant can really help displacing probably C. difficile off the gut. So there are arguments for it. There are in this specific case.
Yeah, it seems like usually probiotics pass through pretty quick and don't hang, the bacteria you're taking in don't hang around in your gut very long. And so, sometimes they'll eat some food on their way through, and that can modify the community. But yeah, I think that once we start doing more probiotics with gut residents, as opposed to things that grow well in yogurt, that then we'll have better luck at getting things to actually engraft and hang out in the gut. And I think that then we may see more of that. So tangentially related, changing the language is one of the things that Linda Kinkel is doing in soils. And I would have predicted the same thing would happen in soils, that you'd mix in a foreign bacteria and it would immediately get out-competed. But she's actually able to show long-term effects of mixing in specific signalers that send out signals which interact with the resident community in ways that lead to suppression of pathogens. So I think there's exciting potential there, but it's definitely challenging to do so far.
Yeah. Are there, if we're thinking about like disrupting communication as a way of sort of stopping sort of the growth or disarming a microbe, are there unintended consequences to that? Or, this may not be going down the right road, but thinking that like the overuse of antibiotics is leading to some serious problems and resistance. And so are people starting to think about, or are they imagining sort of unintended consequences of disrupting some signaling between some particularly bad microbes that would also have consequence on our health and our well-being?
Yeah, I would think that this could happen. It's a language, and so we have to imagine that if we consider that this language is important to a certain process, changes or influence on this language can have beneficial or deleterious effects. And so this is one of the reasons why we really need to study and have very specific tools, which is what my lab is currently working on, where we're putting a lot of effort in generating highly specific tools. So we can alter and modify the presence of signaling molecules in the environment very specifically. It's very important. So I think this is definitely going to be the case, that we will have. Right now, what we find is that because we have disruptors that are very broad spectrum, we find that every time we use them on a biological process involving a complex sequence of events, we find that it disrupts it systematically. And we consider this as a positive effect because we are looking at the process that's negative, right? So let's say biofilm formation or biocorrosion or the death of a worm by infection. We're looking at the process that's negative. When we disrupt communication, we see a beneficial effect because we see a rescue or a prevention of something happening. But in reality, what I think the disruption of communication is doing is simply to disrupt a process that would otherwise reproducibly happen. And so if we were to look at positive scenarios, My guess is that we will also likely see disruptions until we have specific tools in hands.
Yeah, I mean, to my mind, the goal is precision personalized medicine. And I think that disrupting communication, like Mikael is able to do, is much further in that direction than throwing in the napalm of antibiotics. Mm-hmm. And I think that sort of ultimately the development of a whole array of different intervention mechanisms, from introducing microbes, probiotics, to changing nutrients through prebiotics, coupled with some smart use of focused antibiotics and quorum quenching, potentially even phage therapy. I think we're going to want that whole tool set to be able to really do precision management of diverse communities in different contexts.
I think on that is a good note to end on. I'm mindful of the time and your time and our audience's time. Audience, I see you and all of your really good questions out there. I will make sure that I share those with our panelists so that they know what kind of exciting questions you had. And I apologize that we haven't had time to address all of those this evening. So I hope everyone at home can give our speakers a warm round of applause and thankful fingers from where they are, whether that's their desk, their kitchen table, or their couch. So thank you both, Mikael and Will. We really appreciate you being here today. I want to let our audience know to be on the lookout for a brief survey tomorrow. We'd appreciate hearing your feedback about this event. And then I also want to let folks know about a couple more things to be on the lookout for. So I'm giving you a save the date for January 21st. The Bell Museum is excited to host a virtual book launch of Minnesota's Natural Heritage, the second edition, and that will be on January 21st. We will have a conversation with the editors of that book, exploring the definitive work. It's been this second edition is coming out 25 years after the first edition, looking at Minnesota's natural history and ecology. So Minnesota's natural heritage has been updated, expanded, and copiously illustrated to account for profound changes to the state's natural landscape in the past 25 years. We will be posting event information up on the Bell Museum's website very shortly. So, but just hold a save the date for January 21st at 7 p.m. And then, excuse me, I'd also just like to let folks know that we have all of our past probable, possible events, those recordings online. I encourage you to take a look. We talked a little bit about fecal microbe transplants, and we had our first event in the series was about medicinal microbes, and we spent some time with Dr. Mike Sadowski talking about that work. So I would encourage folks to take a look. So I thank you all for joining us tonight. I hope you have a great evening. Stay well, be healthy, and we'll see you next time.